Welcome to the news channel Biology 1110, streamed by LMU Network. Scientists are taking a closer look at one of the tiniest but most diverse, interesting creatures located in freshwater ecosystems. In today's interview, we will dive into these microscopic but mighty animals and highlight their role in their ecosystem. Welcome our three guests today, the Rotifera. So nice to have you guys today. Right off the bat, you guys look pretty diverse from one another. Care to explain?
I'm Deloidia, and today we will each be representing one of the three main classes of Rotifera species out of the 2,200 known species. I asexually reproduce and I live in a freshwater environment. How about you guys?
I'm monogonal. I also live in a freshwater environment, but can be found hanging around in soil environments. My mode of reproduction is both sexual and asexual.
I'm Nadia. My mode of reproduction is also asexual, and I live in a marine environment surrounded by crustaceans.
How interesting is that? Now I'm going to ask you to travel back in time to when you were just an embryo. Can you tell me about your developmental pattern?
As part of the Rotifera species, I am categorized as Lophotrochozoa, which is one of the two major clades of protozoans. A characteristic of protozoans is that their blastopore, or point of invagination of the ectoderm, the outer germ layer, becomes the mouth opening into the alimentary canal. We are also related to mollusks and annelids, a diverse invertebrate phyla within the superphylum Lophotrochozoa.
Thank you for sharing. You're already very fascinating, but let's get serious. What is the value of the Rotifera taxon? How do you connect to us humans?
It can be used as bioindicators of water quality to reflect toxicity of varying solutions because we are sensitive to environmental changes. When studying the presence or changes in our rotifera communities, scientists can get a clear picture of water quality and ecological health.
We are also vital to our ecosystems because we convert algae and bacteria into food source for other organisms.
Thank you. Now, what are the defining characteristics of your species and how do y'all eat?
Rotifera have an extracellular digestive system. We usually eat a lot of bacteria and algae, mostly organic stuff. People think we're small, only unicellular, but we pack a punch. In fact, we are multicellular, almost thousands of cells big, and can grow up to 0.5 milliliters long. Some key characteristics about us are that we have a corona, which is a ciliated crown on the top of our heads, used for feeding and movement. We also have pretty muscular pharynx with jaws. And lastly, we all have pseudocoelomites body cavities.
Tiny but mighty for sure. And how rude of me. I didn't even ask how your trip was. In fact, how did you get here?
Our trip was fantastic. Well, we got here through various forms. We are always migrating. I swam here.
We crawled here.
So, reporter, now that you have our life story, do you know our evolutionary species?
Yes, of course. Let me explain to you. My team and I built a phylogenetic tree and found them from literature to better help understand your phylogeny. First, we have our tree we made in our lab. In the tree, we compared rotifera's characteristics with those of the species we have dissected. We have found that they are most closely related to Echinodermata, and the outgroup is Porifera. This tree is different to the one we found in literature because the data matrix has a much broader range of species. We found this phylogenetic tree from an article published by Acta Zoologica written by Ol Riemann and Wilke Ulrichs. The tree from literature compares other phylum with the Rotifera to see what traits have been evolved over time. The tree splits Rotifera into two, Hemerotifera and Monogonta, then shows other closely related species. The outgroup in this is Plathomynths. Rotiferas have existed for at least 56 million years. And with the trees in mind, we hypothesized that Rotifera originated from an early form of protostome and over time adapted to its microhabitats and diversified by modifying its features.
Wow, you know more about me than I do. That's the diversity that accompanies living organisms like myself.
Thank you for taking time to talk to us. I would say see you later, but I only have a lifespan of 6 to 45 days, so really I may never see you again. Crazy to think I was just in my larval stage 7 days ago.
Well, that's all the time we have for now. Thank you for coming on and giving us some really cool insight. Before we go, I'd like to thank our sponsors, Dr. Roulette, Stephanie Flores, and the Seaver College of Science and Engineering. Tune in next Tuesday at 8 a.m. for our last screening before we go off to break.